Hello, my name is Fischer, and I'm a high school student in this year's SIMI program. My lab mentor's name is Dr. Oman Mattal, and together we work in Dr. Rajesh Khanna's pain pharmacology lab. In our lab, we try to answer this question: how can we regulate chronic pain?
Before I explain the specifics of my lab, I'd like to briefly explain what chronic pain is. Chronic pain is a pathological pain state that persists for months to years after injury has occurred. Currently, there are millions of people in the US that suffer from this condition, and right now our only solution to this issue is the use of opioids. In order to reduce the use of harmful and addictive opioids, our lab is trying to regulate chronic pain by harnessing CRIM2, the collapse and response mediator protein 2.
This protein plays a key role in the communication between neurons, as it directly affects the ion channels of the cells. CRIM2 binds itself to these ion channels, which are located in the cell membrane, allowing communication of pain signals between neurons. Ultimately, we are trying to inhibit changes that occur to CRIM2, such as phosphorylation. Phosphorylation is the addition of phosphoryl groups to the protein, which are chemical ions. In doing so, we hope to regulate chronic pain. It is important to understand that pain should not be eliminated entirely, as it is an essential survival instinct that humans depend on. Therefore, my lab is trying to find ways only to regulate chronic pain to the point where excessive pain is inhibited.
Throughout the summer, I assisted in conducting multiple experiments to provide evidence for our lab's research. One of the most vital types of experiments I have assisted in is Western blotting. Also known as immunoblotting, we use this technique to detect CRIM2 in a sample of tissue extract, and then we analyze the protein and changes made to it, such as phosphorylation. By analyzing these samples, we can gain a better understanding of how these changes affect pain signaling. In addition to this, I assisted in an experimental technique known as calcium imaging. By applying different chemicals to neurons under a microscope, we can observe CRIM2 and its phosphorylation process to see if the chemicals limited communication between neurons.
These chemicals are then used to create pharmaceuticals which can help regulate chronic pain.
Pain is universal, so finding ways to regulate it can help millions of people. Therefore, there are endless clinical implications regarding this research. In conclusion, after conducting our research, some of the questions that arose were: what compounds can be used to limit CRIM2 post-translational modifications such as phosphorylation? What is the most effective way to administer a compound to produce the best results in patients? How can we change our methods of research to collect more accurate results?